Very warm welcome in the Life Science Academy. In this session, we will talking about the DNA replication termination process in detail and mechanism of DNA replication. I have uploaded the part 1 of the DNA replication in which I will explain you initiation and elongation process in detail, and this is the second part of the DNA replication process, and this is the last event in the DNA replication. This is the termination process. So if you have not watched that video, you have to watch that video first and get forward with the DNA termination process. So let's start with the DNA termination process. Termination process in prokaryotes, or you can say E. coli. The model organism in prokaryote is the E. coli. So the circular E. coli chromosome. So there will be the circular E. coli chromosome. So there will be the circular E. coli chromosome. In the circular E. coli chromosome, there will be approximate 245 base pair long sequences present, which we call it as a ORIC, or you can say origin of replication. In the ORIC region, this is the region where the initiation gets started. And if we talk about size of E. coli, the size of E. coli is 4.6 into 10 to the power 6, or you can say 4.6 million base pair long. The chromosome of E. coli 4.6 into 10 to the power 6 million base pair long. So DNA is replicated from a single origin of replication, and that single origin of replication, I have told you, this single origin of replication is called it as a ORIC. Two forks are established, each containing a hexameric replicative helicase. DNA synthesis protein DNAB, which will acting here, is a DNA helicase. So DNAB is a DNA helicase. In the first part, we have told you what is the enzymatic machinery required for the DNA replication process. So DNAB is one of them, enzymatic machinery. So if we have the chromosome, this is the circular chromosome, and this is the region where the ORIC, so the two replication fork move in opposite direction, and these two replicative polymerases are move in the opposite direction, and one is the site where they get meet and fuse with them, and this site is defined as a termination sequence. So two fork are slabs established, each containing a hexameric replicative polymerase, and this replicative polymerase is called the replicative helicase, and this replicative helicase is the DNAB. So each DNAB helicase bind to at least two molecules of DNA polymerase. Third, here the major replicating polymerase enzyme is the DNA polymerase third. DNA polymerase third, which will synthesize, or you can say polymerize, the E. coli's genome. We synthesize the leading and lagging strands in association with the possessivity clamp beta. So clamp beta is like a associator, or you can say acting as an assistant. Suppose we have the structure of, so this is the DNA polymerase and this is the beta clamp. In the beta clamp, suppose we have the DNA, and that DNA will be getting replicate. So the beta clamp, this is acting as a processivity maintainer. You can say processivity is enhanced by the beta clamp of the DNA polymerase third. The two replication fork emanating from a ORIC travel around the chromosome in opposite direction. at a rate of approximate 60 kb per minute. What we said that 60 kb approximate 60 kilo base pair per minute. This is the rate of DNA polymerase third. So, and terminate in a specialized region across from the origin. The termination zone contains 10 dirt sites defined as A to Z, or you can say A, B, C, D up to the Z, which can bind the DNA replication terminus site binding protein, which we call it as a TUS protein, and comprise potent and polar replication fork barriers. The TUS sites are oriented such that the left fork can pass the first 5 TUS sites in encounter, which are marked as red arrows in the upcoming diagram. So, I have explained with the help of a simple short diagram, so you can understand better. So let's forward. So if we have the circular genome, this is the if we have the so if we have the circular genome, so this is the circular genome. If we talk about what happened in this time, so this is the circular genome of E. coli. So if we have the ORIC in this region, we have ORIC. So this is the ORIC region, origin recognition complex. And this ORIC region, we have two opposite directed replication fork, and there will be some sequences which we call it as a to Z sequences present in the So this is the region which we call it as a origin of replication site. So in the site there will be two replication fork present, and these replication fork moving in opposite direction, and where they get meet and fuse, there will be some sequences which we call it as a TER sequence, and these TER sequences are named as A to Z TER sequence. So these TER sequences is acting as a terminator sequences, termini, or you can say terminator sequences. In the terminator sequence, there will be a binding of a protein, namely called it as a TUS protein. The TUS protein bind over here, all termini are bounded with the TUS protein, so making a TER-TUS complex. So there will be formation of TER-TUS complex. When the TER-TUS complex is found, where the TER-TUS complex is found, the DNA replication machinery is understood that this is the site where I want to replicate, where I want to replication terminate. So the function of TER-TUS complex in replication termination remains unclear, but it has been debated whether fork encounter occur after one of the two fork has already stalled or arrest at a TER site, or whether fork encounter occurs between two TER sites. Some fork clearly collide with the non-permissive phase of a TER-TUS complex. So if we have the TER-TUS complex, this TER-TUS complex is acting a barrier to DNA replication process. So in this time, there will be the arrest possesses in the TER-TUS complex, and the DNA B, which we called it as a helicase, is stalled, or you can say fall off from the replication fork, and the DNA polymerase third, which we call it as a polymerizing enzyme, is also fall off from the replication fork. So this is the counterclockwise, and this is the clockwise, not the counter. This is the clockwise and this is the counterclockwise direction of replication fork. When will be the first come at the TER-TUS complex? This is defined by the processivity of the DNA polymerizing enzyme, or we can say both of them, one of them can be eligible to bound with the, or to encounter with the TER-TUS complex. The replication fork can enter but cannot leave when the TER-TUS complex meets the replication fork. We have supposed this region, the replication fork, and this, there will be the another replication. When the replication fork meet, the replication get terminate. The replication get terminates. So we have look like a picture. Suppose we have this type of So this is the tertus complex, and after the termination we have like this. We have two circular chromosomes after the termination process, and this termination is defined by or determined by the tertus complex. So tertus complex have a significant role in the DNA replication termination process. The replication fork can enter but cannot leave, and this is the significance of tertus complex in the DNA replication termination process. So this is the all about the DNA replication termination process, and that will require for the termination of the E. coli's genome. Previously, chromosome 4.6 into 10 to the power 6 million base pair. This is the size, and same size we have two circular chromosome which will possess the same circular genome. This is the termination of the mechanism, or you can say replication process. We have recalled that enzymatic machinery, initiation, elongation, and termination. In the termination, the major role of the tertus sequence in the elongation phase, there will be the role of DNA polymerase third and its processivity maintainer, which we call it as our clamp beta. And in the initiation, we have the DNA B, which will be acting as a helicase that unwind the DNA. These three steps are required to replicate DNA and terminate the DNA replication. Thank you, and please subscribe and share to our channel so that I got multiple reviews for their, or you can say for their particular subject. Thank you.